I'm going to briefly introduce our panelists. Two panelists will set the stage for the discussion, and then we'll ask them to weigh in on their thoughts, sort of on the 3 to 4 pressing questions in this arena. Conversation is going to be really free-flowing, somewhat like a cross-idea exchange, a town hall, and a respectful debate. And we'll pause at 2 different times to conduct the poll with the audience, and then we'll have the panel weigh in on the audience voting and the feedback. And so I just would like to take a moment to recognize our fantastic sponsors, 10x Genomics, Vertex, Prevention Bio, Imsice, and Inspiro. And without their support, we wouldn't be able to bring you State of the Science. You can find more about these innovative companies on our website and at our Ask the Expert series, both live and archived on our YouTube channel. I have a few pro tips for attendees to submit a question. Just place it in the Q&A. You can submit questions throughout the presentation, and we'll answer them questions, as time allows. You can also upvote favorite questions. And again, this event is being recorded live. We'll house it on our YouTube channel after the event for further viewing. So in the interest of saving time for most, for our discussion, I'm gonna do very, very brief introductions for each of our scientists. We could actually take up the whole time discussing each of them. They're valued and integral members of the T1D research field, and you can connect with them on our website in the collab, where many of them are members, or reach out directly. So first, I'd like to introduce Dr. Qizhi Tang. She's at UCSF. A major research focus in the Tang lab is regulatory T-cell therapy for autoimmune diseases and transplantation. Since 2011, she's been co-directing the regulatory T-cell program at UCSF and has supported 10 early phase clinical trials in type 1 diabetes. Her lab's also optimizing islet transplantation to enable highly efficient immunosuppressant-free replacement of pancreatic beta cells for the treatment of type 1. Welcome, Tang. Dr. Todd Brusko, he's at University of Florida. His lab is at the forefront of cellular engineering and genotype-phenotype studies in T1D using high-dimensional single-cell profiling methods. And the efforts of his lab are focusing on normal human immunity, or laying the foundation for subsequent studies mapping pathogenic immune development that leads to disease, including type 1. Welcome, Dr. Brusko. Thank you. We also have Dr. Leonardo Ferreira from MUSC in South Carolina. Is that right? Yes, fascinated by the phenomenon of immune tolerance. His long-term goal is to control how the immune system defines self and non-self, and such knowledge will allow one to design and develop powerful new engineered immune cell therapies to fight autoimmune disease, cancer, and aging. His recent bioarchive preprint is really exciting. Precision engineering of an anti-HLA A2 chimeric antigen receptor in regulatory T cells for transplant immune tolerance. I would encourage people to check it out. Brian Fife, PhD from University of Minnesota. His lab is focused on understanding immune tolerance by studying checkpoint inhibitory pathways, CTLA4 and PD1, PDL1, and more recent peripheral tolerance studies are focused on the use of Tregs. These include chimeric antigen receptor T cells, or CAR T cells. Dr. Levings, Megan Levings, PhD, she's coming to us from Canada at UBC, and her current work is focused on determining how Treg cells differ from normal CD4 T cells at both the biochemical and molecular levels. And her recent publication, Functional Effects of Chimeric Antigen Receptor Co-receptor Signaling Domains in Human Tregs, and Helios is a marker, not a driver, of human Treg stability, say much about her focus. I'm looking to see, I think we may have Alex Marson from UCSF join us. He's also...
I'm here. There's some kind.
He's here. Okay, great. And so his labs, he's coming from UCSF. He's also involved in genome engineered human T cells. And we may also have Georgia Fousteri from San Rafael, Italy. She's also focused on Tregs. Finally, Matthias von Herrath. He comes from the La Jolla Institute. He may pop in for a little bit. And last but definitely not least, Dr. Mark Atkinson from University of Florida. Doesn't need much introduction. He's a giant in the field of type 1 diabetes, and his research is broad in scope, but directed at identifying and preventing a cure for type 1 diabetes. Okay, so thank you all for joining us. This is an amazing group, and I'm hoping that Dr. Brusko and Dr. Tang can set the stage for this discussion, which is reprogramming Tregs and other whole cell tweaks.
Monica, can I share a screen quickly?
Yes, you can.
All right. Well, good morning, everyone, and thank you for joining us. It's really a pleasure to get to see old, familiar friends and faces and colleagues that I've been fortunate to work with for a long time and known for quite a few decades at this point. So let me see if I can share screen now. Okay, here we go. I have one slide, and it's really just a cartoon, and the goal of it is really just to kind of set the stage for what we're talking about here today. So, I think for the most part, we're interested in the role of regulatory T cells as a way to restore tolerance in type 1. And I thought I would set the stage by just actually going back and very briefly looking at the history. Why are we talking about Tregs as a single, subset? Why do we think this is such a critical population? And it goes all the way back to mid 1970s. Gershwin and Kondo basically looked at and found a suppressor population of T cells, and then in 1995, Sakaguchi really identified the first surface marker that could help us pull out Tregs. So CD25 on the surface of Tregs that is constitutively expressed. And then in 2001, FOXP3 was really identified both in mice and in human cells, human Tregs. And there in mice, it creates this profound autoimmune condition. It's called the scurfy mouse. In people, it's referred to as IPEX syndrome. And why this is so important for us as a community and as a group is because it really, there was this observation that patients that had mutations in FOXP3, almost 70% of those patients developed type 1 diabetes, suggesting that this population is really critical for maintaining immune tolerance overall. And then around mid 2000s, we developed protocols to help isolate these Tregs and grow them in vitro to what we think are going to be therapeutic numbers. And then it was sort of work by Tang and many others that are on the line here demonstrated that the specificity of Tregs made a big difference in the overall effectiveness of these populations. So with Alex, who's on the line now, he's really pioneered, I think, protocols for genetically engineering T cells, including Tregs, to change the specificity. So this can be done now by changing the T cell receptor on the surface of Tregs, and we've published some of that work. It can also be modified, and we can take a lesson out of the cancer immunotherapy space and put in these chimeric antigen receptors that may target a tissue directly to antigens that are expressed on the cell surface. And finally, I think there's still additional modifications that we can think of moving forward, and this may include the delivery of other agents using biomaterial type approaches if we have a cell therapy product that we're putting in. And then some of the questions that I think this panel and group should really address is, are Tregs going to be enough? Is it going to be enough to really change the tide of autoimmunity and correct some of those immune regulatory defects? Or what are some of the other combinatorial agents that may be necessary to do that? So I have some of those questions here, but I'll pass off on addressing some of these right now, and I'll turn it over to Tang. So Tang.
Right, thank you so much, Todd. That was a great introduction. I didn't prepare any slides. I thought just to set the stage, we should think about why. And then I have 3 whys. And I think, Todd, you already addressed one of them. Is first in why Tregs. As Todd mentioned, IPEX patient with FOXP3 mutation, that's an experiment in nature in humans. We know that Tregs has a special role in type 1 diabetes. With their deficiency, there's so many organs and tissues, but we saw very high penetrance for type 1 diabetes. It really shows the importance of these cells. And I think the second why is why whole cell? And then why not just simulate the function of Tregs? And should we just give CTLA4 and then or block IL-2? So we have learned, once we discovered the importance of Tregs as a field, we were very eager to learn how Tregs control autoimmunity. And everybody had their favorite mechanism for a while. And then very soon we found, and there were more than a dozen mechanisms. And then the field realized that it's not one mechanism, not two, not three, but a variety of mechanisms that Treg can use in different tissue contexts at a different stage of immune activation to promote immune tolerance and tissue repair. So it's very hard to simulate the function of the whole cell. In fact, the Treg, they themselves can be the smart drug, and they know where to go and know what to do when they encounter the antigen. And then third why I want to mention is why engineering. And Tregs are so wonderful, they're so important for type 1 diabetes, why not just use them? And for that, I think we need to keep in mind that people develop type 1 diabetes or other autoimmune diseases with the presence, most of them have Tregs. And then it shows either their Tregs are not working so well, or their other cells have overcome the suppression and regulation by Tregs. And then therefore we need to help the Tregs, the therapeutic Tregs, to work better in this hostile environment. So we need to engineer. I think many of us on this panel are trying to figure out ways and how to fix the defect that's happening in autoimmune patients, in type 1 diabetes patients. I think once we agree with why Tregs and then why whole cell and then why we need to engineer, and then the question is and how to do it. And I think we'll have 1 hour to talk about that. I'll stop there.
That was great. I mean, really, I think the visual and the way you sort of both framed it is just an excellent starting point. Thank you so much. I just wanted to open it up now to a couple of our panelists, just sort of like, getting their impressions on which cells, which pathways are targets for, in T1D autoimmunity. I wondered if Dr. Levings could answer, and then Dr. Ferreira, Dr. Fousteri, if she's on here, and then Dr. Fife.
Thanks, Monica. Yeah, I think Tang really articulated well why so many groups around the world think Tregs are going to be an important approach to targeting type 1 diabetes. And I just wanted to follow up on something Tang pointed out, which is the multiple mechanisms of action. And one of the things that I've been thinking about recently is how that really is still a black box. And we really don't know which mechanisms are the ones that are important, especially when we start talking about, do they need to be at the tissue? Do they need to be in the lymph node? Do they need to have tissue repair property? So we have this long list of things, and which ones do they need to do? And as we talk about engineering Tregs, trying to enhance particular pathways, it really illustrates the need for more basic science to really know which mechanisms are functioning when and where, so we can optimize those pathways.
Yeah, that's an excellent point that has to be dug into. Dr. Ferreira, do you want to weigh in on that? Or
yeah, I really agree that we need to know more about yeah, can you hear me? Yeah.
Yes, perfect. Sorry.
Yeah, I'll just say I agree that we need more basic biology of Tregs and see what pathways we should really go after. Like in my opinion, there's two that have been on my mind recently. There's the insulin receptor pathway, and there's the IL-2 receptor pathway. Insulin being bad for Tregs, and then other people jump in, and then IL-2 being good for Tregs. And so I think those two, both from basic biology studies and from engineering studies, seem to be two things to look at in Tregs in general, and in specific in type 1 diabetes.
Nice. Dr. Fife?
Yeah, thank you. So I think one of the other pathways, probably why I'm on the call, is the lessons we learned from cancer immunotherapy, looking at programmed death 1 or PD1 and its primary ligand PD-L1. And I think there's a lot of tricks we can learn from the cancer field, and maybe we can, instead of block this pathway to engage the T cells, we need to turn this pathway off on the effectors. And if there's things that we can promote Tregs and able to do that, I think we'll have a, a global effect within the islets, because PD-L1 expression is so critical, I think, to limit T cell function. And it goes up very dramatically with interferon gamma expression. So I think that this is a pathway that we could target to limit effector T cells as well.
I guess let's just sort of turn it towards Dr. Atkinson and Dr. Marson. Is there something we're not factoring in here that we should?
I'll let Alex go first. Go ahead, Alex.
All right, and I apologize for the technical problem with my camera. I think that this has been super well articulated. I can just talk a little bit about the approach that we are taking. We've been developing tools for CRISPR engineering of a number of different T cells, including regulatory T cells, and initially really focused on one particular family that has monogenic mutations that affect the IL-2 receptor and affect Treg function. And the children and young adults in this family that have 2 mutations in this gene have an IPEX-like syndrome. They have Tregs, they're just not working properly. We think that this is an initial entry point to use CRISPR in a clinical context to fix a known mutation and restore Treg function. But I think it also opens the door down the road, if we're successful and develop a protocol for CRISPR engineering Tregs, to start actually engineering and putting synthetic biology into these cells to start going after some of the pathways that have been mentioned by others on this call. So I think one interesting thing will be, in addition to all the critical natural biology that will come out of the basic sciences that Megan and others have mentioned, can we think about synthetic programs that might not actually exist in nature that we can start putting into regulatory T cells to enhance their function and overcome some of these limitations? And I'll just plant one seed that I'm starting to think about is, as we think about cell replacement therapies for the islets, can we actually start engineering regulatory T cells that have a synthetic ability to home to these replacement islet cells and actually enhance their engraftment and function? And so I think this ability to start engineering crosstalk among cells, immune cells and beta cells, is a direction that has enormous.
I'm not sure Georgia has had a chance to speak. Georgia, do you want to say something?
Thank you. I just saw that you had joined the panel.
Yes. Hi. I totally agree with the panelists that spoke previously. Can you hear me all right? Okay. I just would like to add that it has come along through all these studies that the location is very important for the Tregs to function properly. It's not only the numbers and the fact that they may have defects in their functional properties, but also it's important to drive them to the right location. In the case of course, of type 1 diabetes within the islets and the pancreatic draining lymph nodes. So that's also another point we have to take into account when we are engineering, let's say, this Treg product that we want to be efficacious against type 1 diabetes.
It's great. So I'll only add as a summary, again, if I were to form a team to try and bring TRex therapies into type 1, these people on this call would clearly be on it, because they got a great perspective. So thank you all for participating. The one thing I will say is that this has been a 15 to 20 year goal. Things have changed over the years since, and that now we have a greater appreciation of type 1 diabetes being a heterogeneous disease. We have a greater appreciation than ever that age is important. There's a competing market out there in terms of other therapies that are going to hopefully come online. How will this find a place in them? I really like what Alex and Todd said in terms of, is this alone going to be enough, or do you need to add something? Or is there potentially, as Alex inferred, another application that need to be thinking of? So while all of you are creating these great models, I think there is this kind of undertow of these other questions that somewhere along the line needs to be answered. But I'll just say again, I remember Jeff Bluestone saying to me years ago that if we can just find something that works, a lot of these problems will fall by the wayside, from cost to who to treat, when to treat, whatever. That'll just come with time. So I'll take Jeff's optimism here that even though there's some pragmatic issues, if this crew and others maybe on the line contribute, this will be figured out.
Yeah, that's a good way to put it. And I think let's see, did I have a question here in the chat. I'm going to ask it to the group and then have people just jump in. It looks like Matthias von Herrath is on here, and he's asking, as the degree of inflammation in human type 1 is very minor, how do we envision to get enough, let's say, antigen-specific Tregs to the site of inflammation, certainly in the pancreas, where there's very little Tregs there naturally, or PDLN? And I think he's under another name, so I'm going to promote him. Anybody?
I can answer. One of the strategies is, of course, to engineer the antigen specificity of the Tregs and make them insulin specific. So in a way, they will be more prone to migrate to the tissue where there are, the cognate antigen is present. And another way is to actually identify molecules that are overexpressed in the tissue, even though at low levels, and engineer the Tregs in a way that they would express that receptor and be able to home to the tissue, or the draining lymph node, as I said before.
So can I just stop and maybe challenge the panel a little bit more on this, because I think it really is a critical question, and it relates to where do Tregs need to act? And I'd like to hear the opinions of the panel, because I think it really has major implications for whether we are thinking about TCR gene transfer and using native T cell biology and how Tregs typically work, or whether we enter this space of inputting completely synthetic CAR type receptors and other signaling moieties that don't naturally exist. I think you can there's strong advantages to the CAR approach in many respects, but it does change the overall biology of the way Tregs normally work. So I'd just like to hear the opinions of some of those on the panel, and maybe starting with Megan as a start.
Sure. So, I mean, there's no definitive answer to your question. I think we need to do some more basic science, but I guess the work that's been done in transplantation has pretty clearly shown that optimal Tregs need to be at both places, the target tissue and the relevant lymph nodes. And so I think it's likely that in type 1 diabetes, you're going to need to recapitulate that as well. So I guess, as you pointed out, with a CAR, can you achieve that? If you make a CAR specific for your favorite islet antigen, is it going to be enough to get those cells into the lymph node? And we don't know the answer to that question. So I think in my mind, it's really important to pursue these parallel approaches of TCR and CAR in type 1 diabetes to really understand the strengths and limitations of the two systems.
And I can go next. And I think the one thing to remember that the Tregs knows where to go. If there's no inflammation in the pancreas, perhaps the Tregs don't necessarily need to go there. And as some other mentioned, that if we have the antigen specific cells, and they tend to go to the local region where they see antigen, and what's the antigen that will get activated and function within that tissue. It could be in the site of inflammation if there's enough attraction for these cells to go there. Or it could be in the draining lymph node where antigen is deposited naturally. In the absence of autoimmunity and inflammation, that's where Tregs work in the lymphoid tissue to prevent overactivation of cells and that react to self tissue. So that but it also depends on the disease, right? If you're treating type 1 diabetes versus treating transplantation, it's very different. Transplantation by itself is very inflammatory at the time you put tissue in. So therefore, the cells need to go into the transplant site immediately. But in type 1 diabetes, really depend on the degree of inflammation in the tissue, and Tregs knows where to go.
Anyone else want to weigh in on that?
Yeah, I'll just weigh in and add that I agree with Megan and Tang. And I think that, they bring up excellent points. And the only thing I was going to add was that the timing of disease progression is maybe also an important factor to consider. If you're at risk just with the genetics to developing T1D, you're going to, perhaps not need them in the pancreas. But once you have inflammation, as Tang had just pointed out, then I think sites of inflammation are also going to be key to target.
Can I throw in a tangent? Which should be, so Tregs will fight inflammation, but can also engineer them to promote tissue repair, right? So we're saying that place is inflamed, it needs to, then Tregs, if they're inflamed, they don't need to go there, can be the lymph node, but since we're engineering the Tregs anyway, can we boost or add new features to make them also tissue repairers? So we do want them to go to the islets, even if not enough inflammation is being pointed in the chat, but we still want them to go there anyway, because they can promote repair of the destroyed islets.
So almost like a preventative approach if someone's at risk. And that leads me to this other question, like what are the sequential tweaks really in cells and the pathways that might result in the optimal sort of takedown of the autoimmune diabetes in both the prevention and the reversal of the disease, which are really like two different time points in the pathology. So, it's like, how can these Tregs be used in these different parts of the disease, I guess?
Yeah, I think we have to define where exactly what is our starting point, right? If it's treating patients with established disease, we can throw in the most engineered, amazing Treg, and it's not going to help us at all, right? So I think we have to be, very honest and serious about where we stand in terms of the capacity for whatever residual beta cells are there to either regain function, and if there's not sufficient beta cell mass, we have to think about putting in either transplanted tissue or obviously cells that are derived from progenitor cells.
Yeah, it's going to be contextual.
The other key thing is that, it really does, I think we have to consider the burden at which we're going in, whether, how much innate inflammation is there, how many effector cells are there, do we need to create some space, deplete some of that existing burden before we think about putting in a Treg? I think it's a reasonable question.
Just to follow up on that, I think it would be useful for people on this call and other people listening to start thinking how you would combine Treg therapy with the various biologic agents that are now making their way to clinical testing or maybe even approval in type 1 diabetes, because I could definitely foresee that once those biologic drugs are in the game, that we're going to have to figure out how to integrate Treg therapy with those compounds.
Like a teplizumab, right?
Yes, for example. I think there's been very little work on trying to combine Treg therapy with other types of compounds.
I think I'll take one more question, then we'll go to our poll. We have a question from Chin to the panelists. Should we consider the plasticity of Treg to convert into pathogenic Th17 cells, et cetera?
Yeah, I can jump in to address that. I would say definitely that's where we really need to do more research, and then to really define Treg plasticity, whether they, how do we isolate more stable Treg? How do we engineer the cells to ensure their stability, especially when we consider trying to make the Tregs islet antigen specific and get them to go into the islet. If they don't stay as Tregs, and then they will be effector cells and then could cause damage. But I think there's lots of controversies, right? And it's hard to purify Tregs. And a lot of the times we don't even know when it's plastic Treg or they're just impure Tregs, or they're a subset of Tregs. We need to learn that.
Yeah,
I would like to add on this, that it's also a point of the type of Tregs that we start with. So there's a discussion on whether we should start from naive Tregs and how are we going to isolate naive Tregs because they seem to be more stable, for example, or use big cord blood Tregs peripherally derived Tregs, which kind of Tregs also. Because that would also depend, the plasticity would also depend on the starting point, I guess.
We have a lot.
Monica, before you do your question, Matthias has been on the line. Matthias, he's been hearing these discussions. Matthias, is there anything that you've heard in the last number of minutes that you either disagree with or think deserves further emphasis?
Thanks for asking, Mark. No. I think the discussions are very interesting. I think the emphasis that we need to think about combinations, especially with the anti-CD3s that probably or hopefully will hit the market at some time soon, is probably an important argument to create space for Tregs, especially if we don't want to envision a full-fledged metabolic prevention trial as the first step, just because these trials take so long. You could envision testing them in recent onset diabetes, and with that, the concept that was discussed, whether we can make the Tregs in humans safely go through the islets where they're probably needed without them changing their function. There's many questions. What I find particularly exciting though about the field is that we will learn, at least we will learn important rules about the human immunology game, which I think is very important altogether.
Yeah, thank you for that. That's a good summary about the need for sort of dual treatment or more than dual treatment, and it'll be interesting to see how that evolves. I'm going to run this one poll, and then we have a lot of questions sort of piling up. But so here's the poll, and I'm asking the panelists also. Whoops, hold on. I'm asking the panelists to also vote. So the poll is, will Treg therapy alone, even with gene editing, ever be an effective long-term therapy in T1D? And it comes kind of right on the back of the questions we've been talking about, is whether or not that will be the case, and what would be the optimal sort of companion drug to use. And it looks like the audience is agreeing that it's going to be not just Tregs alone that'll do the job. In fact, like 71% of people are saying no. So let's see, I'll get up to, people are voting still. Let's give it a couple more seconds. So almost everyone has voted, 65%. Come on. There we go. Okay, so the end, I'm going to share the results, and it looks like an overwhelming no. And that kind of just comes on the heels of what we were just talking about, that, there's room for other drugs in this treatment, protocol with using Tregs. I'm going to go over here to.
Can I just add? I voted yes for this, and representing the minority. It's not.
Shoot. Alex is in the minority, but he's also hard to hear. But I would like to hear what he says.
We can't hear you, Alex. You want to try again?
I'd be up for having you write it in the chat, and we could read that as well if you want to. Let's see. We'll stop sharing. Well, let's see, maybe he'll write it in. But right now I'm going to go to Nayara Laty's question from Harvard is, in the case of cell replacement therapy, do you think that engineering beta cells is enough to attract Tregs to the transplantation site to avoid autoimmunity, or do we need to use Treg therapy combined? Anybody?
I think it would be a very good area to actually bring TRex therapy rapidly to the clinic, because you could put them, if you transplant stem cell derived islets or just native islets, you could put them at the same site and learn that once you put them right there with the transplant, what these guys are able to do, especially if they have an islet specific receptor. So I like that thought. One thing that we would need to enable for that is that ideally, when the Tregs go in a transplant setting, we don't have full-fledged maybe immune suppression at the same time the Tregs are in, but more like an induction regimen where then the Tregs and other things would be in the maintenance regimen. So that's how I think about it, but that's not an easy question, how immunosuppression associated with transplantation would affect the Tregs. But overall, I think it's very interesting thought and probably clinically feasible.
Yeah, it's part of our second poll, and I'll get to that in a second. It looks like Alice Marson was able to come back in, and he was saying that he was going to argue that there's a promising role for prevention in high-risk individuals. Probably those maybe thinking about people that have, one or two autoantigens already, and that maybe Tregs can have a role with those types of people.
About that. No.
Well, okay.
Yeah, it's
still kind of wonky. Sorry, guys. I'm sorry about that. Let's do the second poll because it's kind of on the heels of what we're talking about. This one is, what alternative diseases might provide the best testing ground for genetically modified Treg as a prelude to testing in T1D? So you have a bunch of different answers, and sorry, multiple sclerosis misspelled, lupus, rheumatoid arthritis, graft versus host disease, scleroderma, psoriasis, solid organ islet transplantation, and other. If you choose other, please join us in the chat and say what your other is. I'm going to let it get up to 50% voting. Interesting. It's really spread across most of them, except for solid organ islet transplantation that has the lead. So let's see. All right, let's take a look. So here's the results, and it looks like solid organ islet transplantation in the lead, 59%. So let's sort of, we talked a little bit about, the beta cell replacement therapy, and now talking about the solid organ islet transplantation as a testing ground. Who'd like to just chat about that a little bit?
I can quickly. I think there's some questions that we should think about as we read through this poll, and one has to do with: are we talking about polyclonal Tregs as a therapy, or are we talking about an organ-specific autoimmune disease where you have a known target, known antigens, and then can you also sample some of that site to see if your Treg cell therapy is actually getting there, impacting the site, and that sort of, those types of questions really, I think, drive what you're thinking around this type of question, whether it's a systemic disease like graft versus host disease, or lupus for that matter, or a much more targeted organ-specific autoimmune disease.
Yeah, that's good commentary, I'd say. Again, all these things have to sort of be defined about, we are talking sort of high level, we haven't gotten totally granular on these things, but it's a good jumping off point for people to start thinking about approaches, I think. I'm going to go to a couple of questions. One from Qizhi Gadir. Is there any study showing mutation in FOXP3? Is it able to induce T1D in B6 or any normal mice? Does anyone know the answer for that?
I was just trying to remember. I mean, I think the scurfy mice die so quickly they don't even have a chance to get type 1 diabetes. That's correct, right? Yeah, they just die of other, I think mostly it's the intestinal inflammation that causes them to die. They can't eat properly.
Okay.
So I guess the answer
Exactly.
to that is, I guess, well, maybe
Yes,
Tang, you were going
they
to
have
follow up.
massive infiltration in the pancreatic islets.
Do they? Okay.
Yeah.
So Ezio Bonifacio just published a nice inducible FOXP3 deletion paper to specifically address that question. And they do, I believe in that paper that was just came out recently, they do see an islet autoimmunity as well in that DTR system.
Okay, on the heels of this, from Boston College, Emra Alton is. He wants to know, in addition to the engineering Tregs in vitro or ex vivo, are there approaches to understanding and prevent the decrease of Tregs in some genetically susceptible subjects and or stop the apoptosis of these cells? Do we know the reason why some patients have less Tregs, and how can we stimulate their proliferation? Is reduced FOXP3 expression happening because of the T1D, or does it cause the T1D? A nice chicken or egg question.
It's a great question, and I think it my thinking of relating to this gets back to many of the autoimmune susceptibility loci that are expressed and really enriched in Tregs. That is what confers overall risk for disease. So, we have CD25, CTLA4, PTPN2 and 22, all of these genes that could collectively impact Tregs, how they traffic, where they function, how they integrate activation signals and negative regulators. And I think, whether or not those cells are participating in autoimmunity is a great and outstanding question in the field. I mean, certainly there have been some animal modelings of that in isolated situations where you have an antigen specific T cell or Treg that once you destabilize FOXP3, they can actually drive disease in that type of setting. Whether that's occurring in human type 1 diabetes, I think is still an outstanding and very important question.
Can I make a comment on top of Todd's with respect to your poll question number 2, Monica, in that I agree. I think antigen specificity is one variable in that list that you gave, but another one, and Matthias may agree with me on this one, is separating them by the type of immune response. Is it an alloimmune response, or importantly, in the case of type 1, is it a relapsing and remitting disease? Because a lot of those diseases you mentioned, MS, RA, they can be relapsing and remitting. Other ones, like that's a very different situation from an allo transplant. And then even in the case of type 1 diabetes, and this goes to Alex's notion about prevention, I agree, it would be very promising, but, it appears in the pre-diabetic, well, at the onset of type 1 diabetes, insulitis is a profound feature. Everybody has it. But in the period beforehand, half the time, even in multiple antibodies positive people, you can go through the pancreas and just not see much, if any, inflammation. So timing may be very important here. But it also depends what the group has said, where this acts. Does it have to act at the level of pancreas, or would you get reprogramming within the lymphoid system? I know that's a lot of comments there, but I think that's another notion that needs to be considered, the type of disease.
Yeah.
That's a very good comment, Mark. That's why I always say that, and we used to have these talks at NPOD. We should still look more into diseases like alopecia and vitiligo. Last but not least, because they are relapsing, remitting, they're heterogeneous, they are localized like PATSI, like type 1, and John Harris and others have shown that we can sample T cells out of the target organ. That's a tremendous opportunity, right? Because in type 1 and many other diseases, we can't just go around sampling what's happening at the site of inflammation.
And John Harris is at UMass, isn't he? Yeah. So maybe we should invite him to one of the upcoming ones, the next series, and talk about this in depth, because that is a really interesting question, that you have access to the skin and you can look at exactly what's happening right in a snapshot type of situation. I noticed that other people added to that list of alternative diseases, IBD, celiac, vitiligo, and of course, alopecia and vitiligo. Okay, that's no problem. And thank you so much, Dr. Atkinson. He's got to run. Thank you again for joining us, but we're going to continue. I have one more question here in the audience. How would it be possible, from Suzanne Pfeiffer, how would it be possible to increase the longevity of administered Tregs? Bluestone tried simultaneous IL-2 administration without success. And we know that Dr. Jeff Bluestone has Sonoma Bio therapeutic company, new biotech company, where he's focusing very heavily on Tregs. So that's interesting. Does anyone like to comment on it?
Let me jump in on that. And first, I think we, when we did the IL-2 plus Treg combination therapy, we actually indeed see an effect of IL-2 prolong the infused Tregs. So there is an effect, but IL-2 is not this specific for the infused Tregs. And one of the challenge we encountered is that they increase other cell types that we don't want to increase. And we have to keep in mind, you know, Tregs are T cells, and then they respond very similarly to other T cells. So try to think of a way to selectively boost Tregs without engineering has been very, very challenging. So one approach is, now that we're engineering, right? Can we engineer a orthogonal IL-2, for example, that only the infused Treg can see? Then you have a selectivity for the infused Tregs to increase their longevity. And then Chris Garcia at Stanford has already developed such a orthogonal pair that can be used.
Anyone else would like to add to that? Dr.
Just very quickly. So another way would be to go from the inside. So I'm a car enthusiast, and so you can have a receptor that includes IL-2 signaling, and the Treg will believe it's always seeing IL-2 every time it gets activated. So it'll just be happier in general, and hopefully increase longevity and stability. So it could be both ways. If we're doing engineering, that I have an orthogonal IL-2, or have a built-in IL-2 signal that the Treg always sees. We ensure that the Treg always has that.
Maybe I can just add to that. So I think both approaches are definitely valid approaches. One thing to think about, I saw there was another question here about potential adverse effects. And of course, the advantage of engineering an orthogonal system is that if there was a danger or safety signal, you could remove the Treg growth factor and get rid of them. So for the car, although you can also engineer things like that in the car construct as well. So it's just thinking about how you would input those safety switches in both contexts.
Talking about adverse events, and I want to add, I think we have experienced about 50 some clinical trials worldwide, and in multiple different indications and hundreds of patients. And then we haven't seen efficacy in most of these early trials. But one thing I think is common across all the trials, it's been very well tolerated, and then there's no overt adverse event can be ascribed to TRex therapy.
Tom, can I ask you a question about the cap? Do you think that there's a limit of Tregs that can be put in or expanded, whether or not it's orthogonal or by just putting in more and more cells? Is there space limitations? Is there an IL-2 limit that will be hit in the body, or what are your thoughts?
That's a great question.
I definitely think there will be a ceiling that you will, the more you put in, you don't get more engrafted. It could be IL-2, it could be, you know, the other quote unquote space, for example, engaging with MHC class 2, sorry. It also depends on the conditioning regimen, right? Roughly there are about 10 to 20 billion Tregs in the human body. And then so you have roughly the idea and how many you can put in and how much impact that will have.
Yeah, we did an experiment in immunodeficient mice where we titrated in Tregs together with a set number of effector cells. This was 100% mouse. And the amazing thing is it didn't matter whether we put in 100 Tregs, 500 or 1 million. After a couple of weeks, they were always at 10%. So it's like the body has this natural rheostat that's I don't know whether it's driven by IL-2 or something else, but there's a maximum number of Tregs that, at least in this immunodeficient mouse setting, can survive.
If you're treating an acute disease, it might be good to just give a lot of Tregs and to reset the system, and then we'll go back to homeostasis and to that 10%, right? But then the work has been done with this dominance of Treg. We do in vitro suppression. We know that it takes one Tregs to 2 T vectors to have effective suppression. So that's about 33% of Tregs. Naturally, you don't get to that level without infusion or without some other intervention. But therapeutically, we could transiently get to that level and then reset the system and then allow to go back to homeostasis.
So Tang, to your point, can I ask another question, which is, you know, we're talking about operating in the T1D space here, right? And we're talking about pediatric kids for the most part, if we want to hit some, you know, considerable portion of patients. You know, there's a lot of amazing things that we can talk about in terms of genetic engineering, but do we need to do all of those things? Can we transiently change specificity? You know, the cancer field has done this effectively and demonstrated that you can transiently put in a T cell receptor or a CAR. Is that a starting point where we could make them antigen specific just for a period of time, get them to the site, clear out some of this inflammation and autoimmunity, and reset the system and maybe push back the continuing loss of beta cells? Leonardo, what are your thoughts?
Yeah, no, that's a good question to clear. I mean, I think in the end, yeah, it's a tough space in general, and then the attention field, because many diseases, like for cancer, we mentioned answer that you're trying to take a page from cancer, but cancer unfortunately really kills. And so people have no choice but to try CAR T cells and PD-1, etc. But in type 1 diabetes space, I think there's so much more care to be taken for the safety point of view. Right? How many cells are going to put in? Are there side effects? Many questions in the chat are, can we combine them, or will they be safe? Can we engineer them? And so I think that's something that goes in the T1D space and not other spaces. So I think that's the first thing, is that for T1D, like, people can inject insulin for the rest of their lives and they stay alive, right? And so I think that that's one thing. The Tregs have to be better than that. And so I think it's something that hopefully with all these different tweaks, right, as the title of the panel is, we can get there. That's kind of a very general thought. But
to reply to Todd's question, I think there is enough evidence in neural models that there is this so-called infectious tolerance mechanism, meaning that the Tregs that we infuse are able to, as I said, to reset the system and induce other cells that can propagate and maintain the tolerogenic effect. So it might be possible. I think it's a very good point, and we should actually clearly test that we could infuse the Tregs and test whether after we remove it, whether tolerance is still maintained. Another question, I had a question for Qizhi. Have you solved the number issue with the antigen-specific Tregs? How many would we need for a patient with type 1 diabetes? Thank you.
Yeah, especially for type 1 diabetes, I think it's really difficult to answer, because when we think about Treg, we think about balance, right? You balance the Tregs with the effector T cells. And then in type 1 diabetes, organ-specific disease, we're talking about antigen-specific, either the antigen-specific Tregs. It's difficult to estimate. I think we talked early on, we talked about heterogeneity of the disease. It might be even different in different patients. I think that's where this reset adjunct therapy will be really helpful, right? You go in and reset and purge out the effector cells and then create the space, and also to allow the therapeutic Treg to be able to dominate. And then that may erase some of these uncertainties of how many cells to infuse.
May I ask, is anyone doing this type of work in non-human primates in terms of the titration of the dose that might be needed to sort of reset or get people to go into sort of remission?
It's hard to do this in non-human primates because there's no type 1 diabetes model in non-human primates. But people have done this for transplantation. It's easier to estimate the frequency of graft reactive cells and then titrate the dose of Tregs. And Angus Thompson, for example, have done this in the transplant setting of infusing increasing dose of Tregs into the non-human primates and
How about
assess
dogs?
its impact.
Dogs do get diabetes, right? Cats. I mean, is that a closer model than the mouse? Or no?
I don't know anybody have grown
Nobody's grown dogs.
So we
T-Rex.
actually considered it for a joint issue, not type 1 diabetes. But yeah.
Yeah, there's not
idea.
strong evidence that pathogenesis in dogs is similar to the pathogenesis in humans, and certainly not in terms of like insulitic pathology, at least.
We just hear a lot of buzz around like how do you translate what you see in the mouse to the human, right? So that's why I asked that sort of naive question.
I think I like to circle back with a few minutes left, and how do we get to humans? And Todd mentioned it's kids and then difficulty, and then engineering Tregs and going in first in humans, and that would be challenging to get into kids. And very early on during this discussion, we talked about how to get to humans, and Alex mentioned, unfortunately, his audio has a challenge, is that maybe we can take extreme phenotype. In type 1 diabetes and with these known genetic problems, right? Tregnopathy, we call them. And it could be FOXP3 defect, and other people are doing this. It could be the CD25 defect that Alex had been mentioned. And then these are, they have extreme phenotype. And maybe, as Brian mentioned, that in cancer and Leo, in cancer patients that can tolerate more risky therapy because of the extreme phenotype. And then if we can select out these patients, and then we can test engineered Tregs in these populations, and that will be the start. And then we will do human experiments instead of finding dogs or monkeys to do this.
I think that's a good idea. I like that a lot because then you're really in a subset of patients who have actual Treg defects and probably multi-organ disease, and you have an easier way to show proof of concept. The other thing, circling back to that, I think for an antigen-specific or CAR Treg, there might be an opportunity, maybe, to put them with these companies that work on islet replacement, right? The Viaside and Semmers and other companies who are working these, and put it in their capsules or together with their islet transplant. But you're an expert on this. How do you feel about this? Because you would get them at the site, especially if there's a capsule, and you would be able to show they're safe. You also would have clinically way out if they're not safe and they destroy the graft, and God knows what happens to them. At least you can cut your losses and say, well, it was all in the capsule. That didn't go very well. So let's try a different modification. How do you feel about that?
Yeah, I think it's really interesting making a composite Treg plus islet graft to treat type 1 diabetes, and especially those that don't have islet function anymore. I mean, Megan has developed this anti-HLA A2 Tregs, right? We can select the graft that express HLA A2. We know it's only on the graft, so it's going to be fairly safe. And we have developed anti-HLA A2. I think that may be an entry point. It's a really interesting idea.
Good. Well, I'm glad we've got some of these really sort of new thought-provoking ideas out there, and we hope to see what you all are going to be bringing to bear in the year ahead. I'm sure it's going to be very interesting. This group is so highly talented and so very well versed in the Treg space, and I can't wait to see what papers are coming up next. So thank you all very, very much for joining and talking to us, and in the interest of everyone's time, which I appreciate the time you've given to this discussion. I'm going to just say we're almost at 9, and I'd like to just sort of announce just briefly that we have the Sugar Science together with DKNet, the Signaling Pathway Project, Endex, HERN, Certain, the T1D Knowledge Portal at UCSD, Protocols IO, AppDears at Mount Sinai, the DRC, and the MMPC are creating this D challenge, which is a bioinformatics based challenge Registration is now open, and the following awards are on offer: up to $20,000 in prize money, a pitch polish session with the JDRF T1D fund, and the DRC an early invitation to submit for their grant. And that registration is open through September 30th. It's a very interesting consensum that the Signaling Pathways Project has put together to look at PBMCs and understand sort of the genetic profile between those with diabetes and not. So I would encourage everyone to try it. It's free. You really have nothing to lose and some things to gain. So give it a shot. Thank you all again for joining us. And thank you for all the incredibly important work you're doing. Have a great rest of your day.
Thanks for hosting, Monica. I enjoyed it.
Everyone, thank you.
Bye.
Bye.